Hey everybody. So these are the answers to who am I? And I really want you to go through the activity and try your best and try your hardest and to do it and then come here hopefully. So please make sure you do the activity. Don't cut yourself short. Try it. It could be challenging at times, but I really want you to think through it. And so who am I is a look at structures of things that we've talked about, the macromolecules we talked about in the last week and a half, and how we can tell which ones are which. And so um, let me go all the way back to the beginning. Sorry. So I'm hoping as a virtual student, you just hit present and you walk through the assignment. If you're an in-person student right now going back through this, that's fine. But try it again. I think this is important. And at the end, um, I hope you learn something. So what I want you guys to do is I want you to tell me what macromolecule, hopefully you did this, tell me what macromolecule I am. Tell me what your proof is. Why am I that macromolecule? And then if you keep trying to add more and more information about the molecule, the better you are off. That's what a lot of biology is. It's about knowing a general fact and going deeper and deeper and deeper and deeper. And I mean, that's what separates like teaching 7th grade bio from 10th grade bio to the AP bio. So we keep adding layers of complexity. So who am I? First guy here, you'll see we're looking for clues who it is. So we say we have 2 rings here. We have nitrogens here. So we have a lot of nitrogens. We have a nitrogen coming off. We have what looks like 5 different carbons. Well, a pentose sugar looks like here. The oxygen base at the point. Got a hydroxyl group down here. And then we have a phosphate coming off. So this is a nucleotide. This is actually adenine. And so it's a nucleic acid, a nucleotide. What my proof is I can see I have a nitrogenous base up here, a lot of nitrogen. I have a sugar, a ribose, and I have a phosphate. And so this will make the backbone. These 2 will make the backbone, and this will be the guy that is the actual information and what changes. Like I said, this is adenine. And so adenine connects to thymine and DNA and connects to uracil and DNA transcription. So with RNA. So that's the first one. As you can see, like I said, I told you who I was. How did you know? What is your proof? And I kept giving you more and more information. Okay. Here, we look here. We have 4 rings. 4 rings are indicative of one thing we talked about in the course of the last week and a half. And as we can see here, before I tell you what it is, there's a lot of carbons and hydrogens. Every one of these points is a carbon. And they have hydrogens attached. Like this guy is a carbon. He's connected with this carbon, this carbon. Carbon likes to be connected to 4 things. So we assume that there's 2 hydrogens coming off too. So there's hundreds of hydrogens, a lot of carbons, not a lot of oxygens. We just have an oxygen here and oxygen here. That means that the chemical formula is going to be like C50H80, 2 oxygens. So this screams lipid. This is a lipid. And more famously, this is a steroid. Steroid always has 4 rings. My proof, a lot of carbons, a lot of hydrogens, not a lot of oxygens, not a lot of polar things. That steroids are always 4. This is actually an anabolic steroid. So this is actually one that somebody would take illegally to build muscle and growth. Anabolic steroids are precursors. Well, the precursor to them is testosterone. So it has the same kind of look at testosterone, but a couple more groups up here, a couple of hydroxyl groups up here that kind of change where they're going to go in the body and what they do. And so this is a steroid. This guy is a little bit confusing. So right now here we have a little bendy kind of shape here to it. Goes in and out, loops, goes in and out again. There's these lines here that kind of give me some hints of what's going on here. We can actually look at the structure inside here. I see that there's R groups. It looks like there is a carboxyl, or was a carboxyl at one point. Now it's a carbonyl, but and I have an amino group. So this is actually a protein. This is the secondary structure of a protein. This is a beta plated sheet. And you can kind of see that it's also making hydrogen bonds across the 2 sides here. That's what these yellow things are right here. And this is a building block of a protein, like an overall protein. So this is the secondary structure of one. What am I? So I'm taking a look here. I'll see that there's a lot of carbons, but there's also a lot of oxygens. There's a lot of hydrogens as well. Um, there looks like there is these oxygen bonding here, so back and forth, back and forth, back and forth. And so we can either say we use oxygen to bond two things. We use it to bind uh, lipids and carbohydrates. And there's way too many oxygens for this to be a lipid. So this is actually a carbohydrate. If you actually counted up all these guys, it's going to be pretty much equal carbons and oxygens in this picture, and double the hydrogens of the carbons. This is actually a mini picture of cellulose, just a little breakdown of cellulose. Cellulose is actually thousands of these glucoses. Each one of these is a glucose connected to itself. And then cellulose wraps on top of itself over and over again in X's, which makes it very, very strong. And so here, a lot of oxygens, a lot of carbons is carbohydrate. This guy kind of looked like a picture at this already before, a little bit different. This guy has two hydroxyls down here, still has the adenine up here, the ribose, but now we have three phosphates. And so this is a nucleic acid again, but this is a different one that we just talked about today. This is ATP. And so we have three phosphates here. It's spring loaded. This one really wants to come off. And when it comes off, it releases a ton of energy. And so that is ATP nucleic acid. Next guy, we have, this is called a chair formation. When we draw things in chair formation, it kind of shows you the 3D formation of this setup. And so when it's pointing up, pointing down, you kind of see like this guy's going down this OH, that guy is going up, these are going directly up, that type of thing. You can see that there's a good amount of carbons. There's also a good amount of oxygens and hydrogens. This is actually a picture of lactose, which is a carbohydrate. Now, I know it's a carbohydrate because, again, the oxygens and carbons are pretty equal. There's a lot of oxygens. There's no nitrogen, so it can't be a protein. It doesn't follow the same amino acid structure that we're familiar with proteins, and it's definitely not a nucleic acid. Now, how I know it's a lactose is a little bit more complicated. I just see that it's two of them, so I know it's a disaccharide, and then I know personally that this is a galactose, that's a glucose, and that's all. But besides that, you kind of see just the general shape here. We at least need to know this is a disaccharide. It's two of them connected together. It's a sugar. All right, this guy, we have a lot of carbon and hydrogens connected. Two oxygens. A lot of carbons and hydrogens connected. This guy is actually a wax, and so this is a hydrophobic wax. The oxygens are slightly polar here, so maybe it could connect to your shoe. And then this guys will make sure that the water doesn't stick to your shoe at all. So this is a typical wax lipid. This guy is also a lipid. It's, again, a lot of carbons and hydrogens. You'll see here there are some oxygens, but this is from the glycerol. And then actually connecting here, this is from the fatty acid here. So this is an ester bond, ester bond, ester bond, so glycerol. This is going to be, we could decide if it's saturated or unsaturated. It's a unsaturated fat. Why that is because it has double bonds. Saturated fats are just single bonds the whole way across. So these double bonds make it unsaturated, and this is going to be a liquid at room temperature. This is a plant oil. Here, we again have a lot of oxygens, a lot of carbon, again, the different kind of structure format, a little bit of a chair formation. You can see that this is coming out of the page. That's what these symbols mean. That's going into the page. And you can kind of see that the N here means that this is repeating over and over and over and over and over and over again. You can see here, we have a new kick into this guy, though. This is odd. We didn't talk a lot about this with carbohydrates. This was supposed to be a little bit of a throw. There's a nitrogen here. Well, carbohydrates are supposed to be CH2O. And so this is a carbohydrate. This is an odd carbohydrate. This is chitin. This is what's used by insects and whatnot as a exoskeleton and for fungi cell walls. This little add-on here adds a little extra strength and like extra spots where they can do hydrogen groups and can make chitin stronger, or does make it stronger. And so this is actually a unique add. This is added through a different process. These are actually glucoses, and then there's a process that happens in organisms that have chitin that adds a amino group and then methyl and a carbonyl group added on to the actual glucose to strengthen it. This guy doesn't give you that much. He gives you a couple, 3 here. So I have looks like an amino acid here, because I can see the nitrogen and I can see carboxyl there, and it also says peptide bond. I know peptide bonds connect proteins, so I know this is a protein. There's just a single strand here. There's no helix, there's no beta pleats or anything like that, no flat pages. So this is definitely just a primary structure of a protein. I can't tell you much else because it's just random pictures here, so it doesn't have a certain job. It's just trying to represent a primary structure of protein. All right, taking a look here. We have a lot of carbons and hydrogens, a lot of carbon hydrogens. Again, we have, it looks like an ester bond, maybe a glycerol here. So this is a fatty acid, this is a fat, this is a lipid. And we could say if it's saturated, unsaturated, this looks pretty saturated, this looks pretty saturated, but then we get a double bond right here. This makes it an unsaturated fat. This is actually a liquid at room temperature, just because that one tiny double bond. Because that one tiny double bond, it acts differently. It's an unsaturated fat. All right, so here I got some phosphates that right away should tell you that it's probably going to be a nucleic acid right off the bat. You can see I have a ribose here, some repeating riboses. So we have the backbone. We actually made a backbone here. And then we have some nucleotides here, here, and here. This is adenine, and so on and so forth going down. Okay. What we're going to see here is RNA. Why it's RNA, not DNA? This is not something we really learned yet, but this is a single strand. There's not a side on over here. If there was one over here matching up, then it would be DNA, but it's just a single strand, one-sided nucleic acid, so it's RNA. Also, it has uracil. This is uracil right here. So DNA doesn't have uracil. Here, this one, I didn't know how to really describe this one or show this one, so I kind of just gave them all. This is protein structure, so this is the primary, secondary, tertiary, quaternary, as we're going along here. You can kind of see here, just like it says, it uses letters that kind of represent a primary structure. These actual letters actually represent an amino acid. We don't really use this lettering system in my class. We kind of just use a 3-word abbreviation, amino acids, but that's because we're not doing long chains like that. Here, we can see the helix, we can see the beta pleats, and then we got them interacting together here in different forms, in different ways, with the R groups and hydrogen bonds and disulfide groups and stuff like that. And then these are a bunch of different tertiaries interacting together. I think you can see this guy is like right here in the middle here, but then we have another guy that's coming over and interacting as well. All right, this guy is a little bit of a trickster. I'm really excited for this one. So we have a lot of carbons and hydrogens. Boop, boop. And we have the glycerol again. So right away, this should tell you it's a lipid. We have double bond here, so your initial guess would probably be it's an unsaturated fat. This actually isn't, and that reason is because of this phosphate up here. So what this actually is this is a phospholipid, what makes up cell membranes. So this is the head of the jellyfish, and these are the tails right here. Okay, this phosphate's extremely polar. This top is kind of relatively polar right here, and this is very non-polar here. So this is a phospholipid, but it's a lipid in its own stuff, but it's phospholipid. And that's it. That's all of them. I hope that was kind of easy. You go back through, maybe take a look at them and see what you guys come up with. And if you need help with the structures, let me know. We'll keep trying to get better at it. All right, guys.